Welcome to AP Biology 1.4 and 1.5. Here we will discuss the properties of biological macromolecules, their structure and function. So far, in our first unit, which is the chemistry of life, we looked at the structure of water and hydrogen bonding, and also the elements that are important for life. And in the last chapter, we had a 10,000-foot overview of biological macromolecules and what their roles are. And in these remaining three chapters of Unit 1, we are going to look at the properties of biological macromolecules, structure and function of biological macromolecules. 1.4 and 1.5 are going to focus on carbohydrates, lipids, and proteins. And this chapter 1.6 is going to specifically focus on nucleic acids. So that is our learning objective, to understand by the end of this conversation, you should be able to understand what makes these four major macromolecules, how they are assembled, and how their assembly sequence is important for their structure and therefore their function. So this you're going to see me repeat again and again. Structure is function in biology. Let's take the example of antibodies and antigens interacting in an immune response, right? So your antibodies must have a specific sequence of amino acids that should give them a specific shape, a specific structure, right? And that structure is important for recognizing the shape of the antigen that it is supposed to bind to. If there was a mutation, for example, in the sequence of the in the amino acids, which causes the structure to be different, then it is no longer able to bind to the antigen. So again, you see the same thing when you're looking at enzymes interacting with their substrates, or how molecules are transported across protein channels across cell membranes, and how gene expression is regulated, and how cells communicate with each other, and how the cell walls of plants and cell membranes of or animal cells are structurally supported and are able to interact with their environment. So again, to give a very broad overview of those four macromolecules, carbohydrates are made up of carbon, hydrogen, and oxygen in the ratio of 1:2:1, and their monosaccharides are their monomers, and glucose is an example of monosaccharide, so is fructose, and they come together to create longer sequences called polysaccharides, examples being starch and glycogen and cellulose, and the linkage is through glycosidic bonds. Lipids are not true polymers, and we discussed that before, and they have multiple roles. They are responsible for energy storage and for signaling and for cell membrane formation and so on. So you commonly find them as fatty acids and triglycerides, and then you also have waxes and steroids, and you have proteins which are made up of amino acid monomers, and they come together to form polypeptides, and polypeptides are joined together using peptide bonds. And you have nucleic acids, which are made up of nucleotides. Nucleic acids include DNA and RNA, and their linkage is through the phosphodiester bonds. So let's look at carbohydrates to begin with. Carbohydrates can include simple and complex carbohydrates. Simple carbohydrates include single sugars, which include glucose, fructose, galactose, for example, and these are the ones that you find mostly in the bloodstream. And then you have disaccharides, which is two sugars coming together that include sucrose, maltose, lactose, and then you have these complex carbohydrates. You may have very short sequences, about 3 to 10 sugars coming together, when you call them as oligosaccharides, and you may have very long sequences, thousands of them coming together, hundreds to thousands of sugars bound to each other, and that could form polysaccharides. Polysaccharides can serve as storage material, as in starch, for example, which are present in plants. The rice, the pasta, the French fries, everything, the potatoes, all of those contain starch, and that is a primary source of energy for a lot of us. And then we have sugars that are stored inside our body, and polysaccharides called as glycogen. And then you have structural formation, structural carbohydrates, cellulose, for example, which make up plants, or chitin, which are present in the shells of insects. And also, a quick note, and this is just more of an interest thing, is carbohydrates can exist in the form of, especially monosaccharides, can exist in the form of sugar alcohols. So you have an OH addition to a monosaccharide, which makes it into a sugar alcohol. And so the examples of those are xylitol, mannitol, sorbitol. And these are what you use in sweeteners, right? So they have a sweet taste to the tongue, but they are not that easily absorbed into the bloodstream. So what that means is you have lesser sugar in your blood. If you're diabetic, that it makes sense to use sweeteners. However, because they are not absorbed as well into the bloodstream, it means that you have an accumulation of that, causing gastric distress and bloating. So you need to make sure that you don't consume these xylitol and mannitol and sorbitol in excess amounts in your system. So carbohydrates, one of the things we need to remember is how they are drawn or represented. The Fisher projection, as you're seeing here, is just a linear representation of how all these carbons are present and where the OH and the H groups are distributed around them. But in a cyclic representation of this, called as a Hawthorne projection, is what shows how the cyclic structure of this sugar is present. So what is important to note here is how we label the numbers of the carbons. So the carbon number 1, which is called as the anomeric carbon, is what would have the carboxyl group in me represented in a Fisher projection. But basically what you need to know is that when you're drawing a structure like this, a cyclical structure like this, you don't even have to write the C here. You just know that at the intersection, each of those intersecting points is a carbon. Okay, so this is a carbon, this is a carbon, this is a carbon here, this carbon, this carbon. So these are all carbons. And so here we have a 6-carbon sugar, and we are showing here that the first carbon is this one, and then second, third, fourth, fifth, and the 6th carbon is presented outside of this cycle. Okay, so this hydroxymethyl group here, this CH2OH, is going to be presented outside of this. Now, why this is important is how you represent or how the OH group is distributed on the plane of this cyclical structure. So imagine this as a plane, right? So we have one plane of these carbon atoms arranged. So if this OH group is below the plane, then you would call it as alpha glucose. Okay, so here is your glucose molecule, but the OH is below here, so it's an alpha glucose. If this OH is above there, then we call it as beta glucose. Why is this important? There's very, very small difference, right? How this OH group is present, whether below the plane or above the plane of the cycle, is going to make a huge difference in the structure. Let me show you how. So we have here alpha glucoses all lined up next to each other. And the first carbon of one of the glucose molecules is bound to the 4th carbon of the next glucose molecule. So that is why we call as alpha carbon, forming a 1, this first carbon, 4, this 4th carbon of the second carbon molecule, linkage, alpha 1,4 linkage. And you have a long sequence of this arranged side by side by side. What happens is there is a structural constraint that is formed. So because of that, this long stretch kind of turns into a helical shape like this. It goes into a helical structure. So we will call this as an alpha helical structure. Okay. And you have also 5-carbon sugars. If we looked at glucose, we also have 5-carbon sugars. An example of this is this ribose here, where we have carbons 1, 2, 3, 4, and the fifth carbon is projected outside. This ribose is going to be so important when you're studying DNA and RNA, because they have ribose sugars, okay, and deoxyribose sugars. It's whether, depending on whether it is RNA or DNA, you'll have ribose sugars or deoxyribose sugars. So these are all pentose sugars, meaning 5 carbon sugars, and these are called hexose sugars because they have, hex means 6, right? So 6 carbon sugars. So we said carbohydrates can serve as energy storage polysaccharides, or they can help in support of cell walls and such. So here we have starch, for example. Starch has 2 types, amylose and amylopectin. And if it has alpha glucose, and all of those are joined by those 1,4 linkages that we looked at in the previous section, and it forms these helical structures, then we end up with the type of starch called amylose. On the other hand, if you have both 1,4 as well as 1,6 linkages in alpha glucose, so what we're saying is that the first carbon and the fourth carbon can link, but then the first carbon and the sixth carbon of another sugar can also link, which means that it's kind of branching off. Right? 1,4, 1,4, 1,4 is just one straight line, but then because the sixth carbon of another sugar is able to come and bind to here, now we are able to form a branch. So this branching leads to a completely different structure of starch called amylopectin. So that's the difference between amylose and amylopectin. Then you have storage in animals called glycogen, again made of alpha glucose, but also having, like amylopectin, it has got 1 and 4. And it forms much more complex branching off structures. So these are just different ways that organisms have chosen to store their complex polysaccharides or complex carbohydrates. But what if you just don't have any branching, and you end up with very long linear structures, as happens in cellulose? So cellulose has beta glucose. So as I mentioned, beta glucose here. So we had alpha glucose where this OH was down here, right? What if that OH was above here? And so the kind of bonds that it is able to form, the form of kind of glycosidic bonds it's able to form, is different. So that simple difference, like I said, that whether the OH is above or below, can create a modification that looks like this, which leads to this really long stretches of glucose molecules. And all of these long stretches of glucose molecules are packed next to each other, and they are bound to each other with hydrogen bonds. And that leads to a certain level of strength, structural strength, which is what plants really want, okay, for their cell walls and such. There's also a type of polysaccharide called chitin. Now, chitin, instead of using glucose, it has a modified version of that monosaccharide as a monomer. And that's called as an N-acetyl glucosamine. Essentially, think of a glucose molecule. You just add this NHCO group, just this little addition here. Now it has become a variation of glucose called N-acetyl glucosamine. And that forms a long chain. And that's called as, then that forms a structure called as chitin. Chitin is what you see as the shell of insects. So, now it's cicada season here in Texas, right? In Dallas. So you see when you walk along all these shells of insects that are on the walls and on the floor, all of that is made of chitin. Okay. So that's what we want to know for carbohydrates. And if there's more information, we can definitely cover them in class. And then we have lipids. So lipids are a diverse group of hydrophobic molecules. We looked at what polar molecules and non-polar molecules are. If you remember very quickly, polar molecules are those that have partial positive or negative charges, and non-polar molecules do not have any partial charges, so they are not charged. And when you have a molecule that has absolutely no charge on it, it doesn't want to work with water because water is a polar molecule. So when we have a molecule that doesn't want to work with water, we call it as hydrophobic, meaning that it doesn't like water. It doesn't like to work with water. It's not afraid of water. It doesn't like to work with water. Water and oil don't mix, right? So lipids include steroids, oils, fats, and waxes. One of the most common type of lipids is a triglyceride. A triglyceride has three, the name tri is three, right? So it's got three fatty acids chains. What are fatty acids? Fatty acids are simply long chains of CH3, CH3, CH3. So CH3, CH2 chains, which are all non-polar. So they have, and finally they all end up with a COOH group or something. So then you have fatty acid 1, fatty acid 2, fatty acid 3. These three fatty acids will come together and bind to a glycerol molecule. Okay, so I think I have its structure here. So you have fatty acids, long chain of CH2, 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 CH3, CH3, and then ending with the COOH group. This is one fatty acid. This is another fatty acid. This is third fatty acid. And these fatty acids are coming and attaching to a glycerol molecule. A glycerol molecule is like a E. This is how I remember it, like an uppercase E, right? So this is like an uppercase E. And you have these three fatty acids coming and attached to one arm each of the E. So now you've ended up with a triglyceride. Okay. So these are the most common types of fats, common types of lipids. Then you also have steroids. Steroids are a little bit different in the sense they have ringed structures. They are four ringed structures, four rings of carbon, hydrogen, and a little bit of oxygen mostly. And so they have a lot of roles in cellular membrane flexibility, and also how they act as precursors for making hormones. So fatty acids can include again triglycerides and a variation of this which is called phospholipid, and then we have waxes and certain variations of these called sphingolipids. Don't have to worry about them. They are present in brains usually. And then you have glycolipids, which are lipids that have interacted with carbohydrates. And again, they have a very important role in your brain function. So one of the things we need to remember about lipids is when you're looking at the fatty acids, right, these long stretches of CH3, CH2 and so on, if they don't have any double bond throughout, then you'd call them as saturated fatty acids, meaning that they are saturated with hydrogen. But if you have a double bond, just one double bond in that whole stretch of a fatty acid, then you'd call it as mono unsaturated. So this is one place, mono, which means one, where there is hydrogen is not fully saturated, mono unsaturated fatty acid. Or you could have multiple places where you have double bonds. Just if you forget what a double bond is, it is where you have two pairs of electrons being shared. So you have a polyunsaturated fatty acid here. Now, when you have just when you have a saturated fatty acid, you just have long chains of this, which means that they are able to pack next to each other. There is no hindrance, steric hindrance, no spatial hindrance for them to kind of rest next to each other. So they all pack next to each other. So at room temperature, that leads to them becoming solid. Examples of butter and lard, and usually they are produced by animals. Now the problem with this, when you're consuming a lot of butter and lard and so on, and if they are not fully digested in your system, and if they accumulate in your bloodstream, is that they can clump into solids. And if they are not cleared, then they can block the flow of blood. And if they block the flow of blood in your brain, that's a stroke. If it can cause a stroke in your heart. And that's why we need to become careful about what we consume in terms of fatty red meat. But when you have either a single double bond or multiple double bonds, that causes a certain kink in the shape. Okay, so when and that means when you have, like for example, if this double bond is here and it is kinked, let me see if there's a structure picture here. So you have, let's say, this is a saturated fatty acid, it's straight, but you have an unsaturated fatty acid. That double bond is not very flexible, so it causes it to kink a little bit. Imagine you're trying to pack a whole lot of these next to each other. Because it is kinked, then it's not able to pack together as tightly as the saturated fatty acid. Because of that, tends to remain liquid in room temperature, which is the form of oils. Plants tend to have a lot of unsaturated fatty acids. And therefore, it is actually good for you. When you have unsaturated fatty acids, avocado oil and things like that, they are good for you. Okay, so one of the things that we looked at was cis versus trans. Now, you remember cis was when you have next to a bond, you have two molecules present in the same side. Then it is cis. Cis means same. Or if it is present in the opposite side, then it means trans. So here I have a unsaturated fatty acid, and I have a double bond here, and I have the two hydrogens present on the same side. So that's a cis molecule. Now I have a trans molecule, okay, where it's present on the opposite sides. So when you see cis and trans in lipids, that's something you should visually be able to remember. Now, let's take a situation where I've taken a triglyceride. Remember a glycerol molecule with these three fatty acids here. I've taken a triglyceride and I've removed this, one of those fatty acids. And in that place, I've added a phosphate group. Now, I've ended up with a phospholipid. Now, this phospholipid has a very important role in forming cell membranes. So, for example, here I have a glycerol molecule, and I have these slightly colored tails, which is the fatty acids, and then one of them is going to have the phosphate group, and that phosphate group is going to become the hydrophilic end because it is charged, and therefore it can interact with water. Okay, so you have your glycerol here and you have your phosphate group. Now it can interact with other materials, so it can interact with water and such. So this is the head of the phospholipid, and this portion which cannot interact with water is hydrophobic, and that becomes the tail. So if you look at how a cell membrane is organized, you'll see all the phospholipid heads are arranged on the top. They all clump together, and all the tails are clumped together in the center. And then you have two layers of that making a bilayer sheet. We'll see this in more detail when we are studying phospholipid cell membranes later on. But that's a very important class of lipids. The next group is steroids. Steroids, as we mentioned, have cycles. Okay, so four cycles of not cycles, but four cyclic groups, and so for example you have here. So we have cholesterol. Cholesterol has the structure, and it has a major role to play in how your cell membrane's flexibility is. So if you look at your cell membrane, you have these cholesterol molecules kind of present in this, and that means that whenever a cholesterol molecule is present, the bilayer is not, the cell membrane is not tightly packed. It allows to be a little bit loose. Okay, so the more cholesterol is present, the more flexibility there is at room temperature, and but of course it has to be in a certain balance. And so that's where most of a lot of the cholesterol is present, and it's also present in the brain in very large quantities. Um, but cholesterol is also important for making hormones. So these here, estrogen and testosterone, they are all derived from cholesterol. And so I want to ask you a question to think about. Why do you think there could be a correlation between eating a high-fat diet and early puberty? Okay, so the answer is probably obvious, but just think through that. Plants also have a version of this called phytosterols. A version of steroids called phytosterols. And believe it or not, plants have hormones as well, auxins and gibberellins, and they are all steroids. Then we are moving on to proteins. Proteins are, as we saw, made up of amino acids. Amino acids have the structure where you have a central carbon and you have an H on one side, you have a carboxyl group on one side, and amino group on one side. So this is all common for every amino acid. And then it's got this R group or the side chain. And the side chain can be different. It can be just a H, it can be a CH3, it can be a CH3CH2OH, it can be a cyclic compound, it can be a polar molecule, a non-polar molecule, whatever, right? So depending on what that R group or the side chain is, you get the unique property for that amino acid. And you have 20 common amino acids that you find in 20 to 21. People kind of struggle between that, but 20 to 21 types of amino acids that you commonly find in our human systems. So when you have two amino acids that need to come together, you have a dehydration synthesis. So you have the OH group and the H group being lost, and therefore you end up with a covalent bond, which we call as an amide linkage, or a peptide bond. And so now you have a sequence of two amino acids coming together. That's a dipeptide. And you can have more and more and more of these attached to each other to create a long sequence of peptides. And that is what makes proteins. Now, this is so important to remember when you're thinking about how DNA, the sequence of DNA is converted into specific proteins. This ability of peptide bond formation to attach one after the other in this long sequence, it's going to be really critical for us to know this and remember this. So I've just repeated this here. You have your amino side of your amino acid, and then your carboxyl side of your amino acid. And so we tend to call this side as the N group, or the N side, N terminus. And then the one that has the carboxyl side is called the C terminus. Not in a single amino acid, but when we have a long sequence of amino acids joined together. Okay, so here I have, using this dehydration synthesis, I have a whole lot of amino acids that are kind of formed to come together. So this first amino acid had this amino acid and loose, okay, free amino acid. That's called a free amino acid. So this free end of the that has the N terminus, the amino group, is called as the N terminus. As this side, you have the COOH group. This amino acid here has the COOH that is free. Okay, so that's called as the C terminus. So this is what orientation is in biology. Directionality is in biology. So you have one side that has a specific role, and the other side that has a specific role because of what free terminus it has. And then when you have a long stretch of amino acids coming together, depending on which amino acid sequences are, what the sequence of the amino acids are, you can have a specific type of folding. Like for example, you have two amino acids here that are able to form that have polar ends. Okay, so these are the side group is a polar group here, the side group is a polar group here. So when they are coming next to each other, they're able to form a hydrogen bond. That's going to stabilize this polypeptide here. Or you could have a particular amino acid that has a sulfhydryl group, sorry, SH group. So cysteine, the name of the amino acid there is cysteine. So cysteine here and cysteine here come together, loses a hydrogen, and forms a very stable bond called a disulfide linkage. So those disulfide linkages have brought these two this polypeptide chain together in this particular shape. Then you have hydrophobic interactions, where you have a group of an hydrophobic R group here and a hydrophobic R group here. They are coming next to each other, and remember that non-polar and non-polar can tolerate each other, and they have some kind of a mild attraction for interaction between them, not attraction but interaction between them. So they are held together. But if you had a hydrophilic group here and a hydrophobic group here, they are not going to work with each other, right? So they are going to push away from each other. And then you have acidic and basic amino acids, and those that have COOH group and NH2 group, they are going to form interactions with each other by giving hydrogens and receiving hydrogens and then forming ionic bonds as a result of that. So that's why, again, sequence is important for structure in proteins. So I'm showing these are the same images, this and this, but just wanted to show that here. So you have a sequence of amino acids. Each of those amino acids has a different type of R group. Depending on the type of the R group, you are able to form either structures that can coil into a structure called as alpha helix, or they can form pleats like a harmonium, right? Like an accordion. So it forms these structures like this. They go up and down like this. So that is called as a beta pleated sheet. And these structures, when you have the alpha helical structure or the beta plated structures, they are called as secondary protein structures. Now the secondary structure can fold again to form more complex structures called tertiary structures. So here we have alpha helices and beta pleated sheets in this very long sequence of protein and or a very long sequence of amino acids. And what these do is they come together attached by hydrophobic and hydrophilic interactions and hydrogen bonds and van der Waals forces and all of those, and they form these structures. So this is a three-dimensional unit. And these, let's say there are multiple proteins or multiple structures, tertiary structures, that need to interact with each other to create a functional protein. That is called as a quaternary structure. So quaternary structure is multiple subunits of proteins coming together to create one functional protein. And you see this a lot as well. So here I have indicated which you don't need to remember, the sequence of these amino acids, which is what. You don't need to remember all of that. Just know that there are different types. You know that there are non-polar molecules like this. There's alanine, for example. It's a CH3 group. The electrons between carbon and hydrogen are equally shared between carbon and hydrogen, and therefore there is no charge built up here. And so CH3 is a non-polar group. So you have a non-polar group attached as the R group in this amino acid. That makes this amino acid a non-polar molecule. So you have other things like this. And you also have these aromatic compounds or ring structures, right? So like we saw with steroids. So tryptophan, phenylalanine, tyrosine, they are all aromatic amino acids. And you have polar molecules. Like here you have a OH group, CH2OH group. So this is a OH group, as we saw, O and H. They have a unequal distribution of electrons, which makes them polar, which means the amino acid that is attached to it becomes a polar amino acid. So the tyrosine here, even though it is an aromatic group, has a OH group attached to it. Therefore, this is a polar molecule. So polar amino acid. So all of these are examples of polar molecules. Polar and non-polar are not going to attract. Polar and polar are going to work with each other. Non-polar and non-polar can tolerate each other. Then you have amino acids that have charges built on them. Like you have your this amino acid here called lysine. It's got an NH3 group. Remember, NH3 is able to give electrons, right? So what that means is it is able to act as a base. And then you have aspartic acid, glutamic acid. They have COOH, right? So the COO group, this one here, and this one here, the H is able to give off hydrogen, making it acidic. So this and this are going to attract to each other, okay, because they will form ionic bonds. So now you can see why all the side chains are so important, and the side chains make all the difference here. When you have these structures, these secondary structures and tertiary structures, they can be disrupted by application of heat or being in a different pH. So all of these structures are maintained at a particular temperature and a particular pH. If the pH is different, for example, what would happen to the polar interactions? Okay, so that's going to be disrupted, right? And what's going to happen to the ionic charges? They are going to be disrupted. And that means that your shape is going to be disrupted as well. And therefore, your protein is not going to function as expected. Same with heat. Heat is going to break all kinds of bonds between molecules. Hydrogen bonds are going to be broken. Your disulfide bonds will be broken. Everything is going to be broken, right? So that's going to cause your protein structure to be lost if you heat it high enough. So when you see egg, okay, so you take an egg and you break it on a hot skillet. The reason why it turns white is because the protein structure is lost. It's in what do you call that? It's lost forever. Okay. Finally, with nucleic acids. Nucleic acids we are not going to study in great detail here, but I'll just give a quick introduction because next chapter is going to be all about nucleic acids. Nucleic acids are information molecules. DNA and RNA are the two major nucleic acids. The structure of DNA determines the protein sequence and therefore it is responsible for determining what kind of protein sequences and therefore what kind of protein structures are going to be formed. So this is called as a central dogma of biology, where DNA uses carries the information and uses RNA as a messenger, and using RNA for translation and so on is able to determine the particular sequence of amino acids that will be present. And so, for example, if I had a particular sequence that told me that I'm going to make a particular type of protein called melanin, and if my sequence was that it would produce a lot of melanin, okay, there'll be some control sequences here. We'll talk about that later. That if I produce a lot of melanin, I'm going to be really dark. If I'm going to be producing a little of this, then I'm going to be white or not so dark. And that means that's all there is, the difference in skin color. Some people are brown, some people are darker and black, and some people are lighter. So again, this is where biology meets life. This little difference in color, why should that matter so much in how we treat each other? We know we are all good people. We don't do that. But I'm just saying, as humans, why we make so much of distinctions between colors and such, when all there is how the sequence of those amino acids are determined by how those sequences of nucleotides are. So anyway, just something to think about. Anyway, I want to end with this really interesting, cool article about how DNA is the storage molecule. I mean, the storage molecule. So here's a really cool article from Scientific American that I would encourage you to read. Humanity by 2025 will generate about 33 zettabytes of data. That's 3.3 followed by 22 zeros. Okay, that's a lot. And you can take all of that information, and if you were to store it in the way that DNA stores information, you could take all of the information that humanity would have generated and condense it into the size of a ping pong ball with room to spare. If you take all the information in the Library of Congress and you could squeeze that into the size of a poppy seed, and you can do that 6,000 times within the size of a poppy seed. That's as much information that you can store inside, stored with DNA. Okay, so this is really, really cool, and the perfection with which information is transmitted, so little error, considering how much of information that is, it's just mind blowing. Anyway, that was our intro to Arvind. That was our discussion of proteins, lipids, and carbohydrates. Hopefully, this was helpful, and hopefully, you have met all of your learning targets on this one, including how the sequence becomes important for structure, and how structure becomes important for function, and how directionality becomes important when you say N terminus and C terminus in proteins, and how we have in DNA, we're going to see this in the next one, how we have 5' and 3' ends, how directionality becomes important in biology. Anyway, hopefully this was all helpful. Thank you so much for listening.